Special thanks to London Heartbeats Z Academy UK for educational support and request listeners to explore its greater than 200 popular courses. Subscribe social media as Facebook, Instagram, YouTube for free regular updates and not miss future amazing discussions. Subscribe to higher level of YouTube membership for early access to educational videos and also even to have personal chats and guidance sessions. Anatomy and Physiology explores how different cells, such as bone, nerve, muscle, and red blood cells, contribute to the structure and function of the human body. We will review key topics, including the cell, integumentary, musculoskeletal, nervous, endocrine, cardiovascular and hematologic, gastrointestinal, urinary/fluids and electrolytes, and reproductive systems. Let's begin with the cell, the basic structural and functional unit of the body. The cell performs many vital functions, including protection and support, movement, communication, metabolism and energy release, and inheritance. The cell is composed of the cytoplasm, cell membrane, organelles, the nucleus, and inclusions. This detailed diagram illustrates the complex structure of a cell and its various components, from the nucleus to the organelles and the cell membrane. The cytoplasm is the viscous, translucent, watery material where the various organelles are located within the cell. The cell membrane is a semi-permeable boundary, separating cellular structures from the external environment. This structure allows the cell to regulate what enters and exits. Major organelles include the nucleus, the rough and smooth endoplasmic reticulum, the Golgi Apparatus, and the mitochondria. The cell membrane is selectively permeable, featuring bilipid layers. Its primary function is to regulate the passage of substances into and out of the cell. Key cell membrane processes include phagocytosis, or cell eating, pinocytosis, or cell drinking, endocytosis, or cell engulfment, and exocytosis, or cell excretion. This diagram illustrates the process of endocytosis, where molecules are transported across the membrane and enclosed in a vesicle. Exocytosis is the process where a secretory vesicle fuses with the cell membrane to release its contents outside the cell. Cell connections include tight junctions, which bind cells together and regulate permeability, desmosomes, which provide a mechanical link, and gap junctions, small channels for ion and molecule passage. This image shows the structures responsible for cell connections, including tight junctions, desmosomes, and gap junctions. Cellular organelles are the metabolic units within the cell, each with specific functions essential for maintaining cell life. These organelles include the mitochondrion, endoplasmic reticulum, ribosomes, Golgi apparatus, lysosomes, peroxisomes, cytoskeleton, and centrosomes. The mitochondrion is known as the powerhouse of the cell, containing enzymes and complexes vital for ATP production. It also contains mitochondrial DNA and is the site of key metabolic processes like the Krebs cycle and beta oxidation of fats. This diagram provides a visual representation of the mitochondrion, highlighting its outer and inner membranes, intermembrane space, matrix, cristae, and enzymes. The endoplasmic reticulum is an extensive network of membrane-enclosed tubules. There are two types, rough and smooth endoplasmic reticulum. Their ER is covered with ribosomes and is the site of protein synthesis. The smooth ER lacks ribosomes and is the primary site of lipid synthesis. This image displays the structure of the endoplasmic reticulum, demonstrating both the rough and smooth components and their proximity to the nucleus. Ribosomes are small structures composed of ribosomal RNA and proteins, serving as the site for protein synthesis within the cell. The Golgi apparatus modifies, packages, and distributes proteins and lipids synthesized in the ER for secretion or use within the cell. This diagram illustrates the structure and function of the Golgi apparatus in processing and packaging materials in vesicles. Lysosomes contain hydrolytic enzymes and function as the cell's waste disposal system by digesting foreign material and worn-out cellular components. Peroxisomes are smaller vesicles containing enzymes that break down fatty acids and amino acids, often producing hydrogen peroxide as a byproduct. The cytoskeleton provides structural support, maintaining cell shape, and enabling cell movement. It consists of microtubules, microfilaments, and intermediate filaments. Centrosomes are vital for cell division, composed of two centrioles. They form the mitotic spindle during mitosis. The nucleus is the control center of the cell, housing the cell's genetic material in the form of DNA. Key components of the nucleus include the nuclear envelope, nucleolus, and chromatin, which is the complex of DNA and proteins. This image highlights the structure of the nucleus, showing the nuclear envelope, chromatin, nucleolus, and nuclear pores. Cellular movement involves cilia, which are short, hair-like projections, and flagella, which are longer, whip-like tails. Both are essential for moving substances across the cell's surface or propelling the cell itself. Genes are functional units of DNA, encoding the sequence for a specific protein. Chromosomes are highly condensed strands of DNA. The process of DNA replication ensures that each daughter cell receives a complete set of genetic instructions before cell division. The genetic code is read in codons, which are 3 base sequences that specify an amino acid, forming the basis of protein synthesis. Protein synthesis occurs in 2 major steps: transcription and translation. Transcription is the process where a DNA strand is copied into messenger RNA, or mRNA, in the nucleus. Translation then uses the mRNA to synthesize a polypeptide chain on the ribosome in the cytoplasm. Cell division, or mitosis, results in 2 genetically identical diploid daughter cells. It is essential for growth and tissue repair. The phases of mitosis are prophase, metaphase, anaphase, and telophase, followed by cytokinesis, the division of the cytoplasm. This diagram illustrates the sequential stages of mitosis, from prophase to telophase, and the resulting daughter cells. Let's review cellular division. Meiosis is a reduction division that occurs in sex cells. Sex cells have a haploid number, meaning they have only 1 pair of chromosomes, which is 23. We will now discuss cell physiology, focusing on how substances move across cell membranes. This diagram illustrates the transport of different molecules and ions across the cell membrane, often aided by membrane channels based on concentration gradients. Diffusion is the movement of solutes or particles from a higher concentration to a lower concentration. This is a passive process that does not require any energy. Here we see an example of diffusion, where salt particles in distilled water gradually spread out until they are evenly distributed. Osmosis is the movement of a solvent, typically water, from a diluted solution into a more concentrated solution through a semi-permeable membrane. The pressure that draws water into the more concentrated vessel is called osmotic pressure. This image shows how osmosis works in a lab setting, demonstrating the movement of water across a selectively permeable membrane towards a higher salt concentration. Factors that affect diffusion include the concentration gradient, particle size, solubility, and temperature. A special type of osmotic pressure is exerted by the proteins in the plasma. It is referred to as oncotic pressure. Filtration is the movement of both solute and solvent due to hydrostatic pressure, moving from an area of higher pressure to an area of lower pressure. An example is urine formation. Hydrostatic pressure is the force exerted by a fluid against its container. Increased hydrostatic pressure is one mechanism that can lead to edema. Active transport is the movement of solutes across a membrane from a lower concentration to a higher concentration, requiring energy. Examples include the sodium-potassium pump, endocytosis, and exocytosis. This diagram illustrates the sodium-potassium exchange pump and carrier molecules, facilitating active transport of substances like glucose across the cell membrane. Here we see the effect of different solutions on a red blood cell. Hypotonic solution causes swelling, isotonic solution maintains balance, and hypertonic solution causes the cell to shrink. A tissue is a group of cells with similar structure and function. The 4 basic types of tissue are epithelial, connective, muscle, and nervous. Focusing on body tissues, epithelium forms the lining, covering, and glandular tissues of the body. Its key functions include protection, absorption, filtration, and secretion of substances. Epithelial tissues are categorized into simple and stratified epithelia. Simple epithelium is lined by a single layer of cells, while stratified epithelium is lined by many layers of cells. Simple epithelia include simple squamous found in alveoli and blood vessels, simple cuboidal in glands, simple columnar in the GI tract, and pseudostratified epithelium in the bronchial lining. Stratified epithelium includes stratified squamous like the skin, stratified cuboidal in the reproductive duct, and transitional epithelium found in the bladder and ureter. This image displays illustrations and micrographs of simple squamous epithelium, which lines structures like the air sacs of the lungs, and simple cuboidal epithelium, which forms structures such as kidney tubules. This slide provides examples of simple columnar epithelium, often found in the digestive tract, and pseudostratified ciliated columnar epithelium, which is common in the respiratory tract. The image here illustrates different types of stratified epithelium, including stratified squamous epithelium, a protective layer in areas like the skin. This diagram further details stratified cuboidal epithelium and transitional epithelium, which is highly distensible, lining organs such as the urinary bladder. Connective tissue is the most abundant tissue in the body. Its functions include supporting and protecting organs, storing fat, and providing immunity. Connective tissues are divided into 3 types: loose, dense, and specialized. Loose and dense are further classified as fibrous tissues. Loose connective tissue includes areolar tissue, adipose tissue for fat storage, and reticular tissue, which forms a supporting framework. Dense connective tissue is classified as dense regular, found in tendons and ligaments, and dense irregular, found in the dermis of the skin. The specialized connective tissues include cartilage, which can be hyaline, elastic, or fibrocartilage, bone, and blood. Muscle tissue is essential for movement and is classified into 3 types: skeletal, cardiac, and smooth muscle. Nervous tissue, found in the brain, spinal cord, and nerves, is composed of neurons and neuroglia. Neurons transmit impulses, while neuroglia protect and support the neurons. The central nervous system, or CNS, includes the brain and spinal cord, which are responsible for integrating information and coordinating activity. The peripheral nervous system, or PNS, is composed of cranial and spinal nerves. These nerves connect the CNS to the rest of the body. This diagram illustrates the organization of the nervous system, differentiating between the central and peripheral divisions. The PNS is divided into the somatic nervous system, which controls voluntary movements, and the autonomic nervous system, which regulates involuntary functions. The autonomic nervous system, or ANS, is further broken down into the sympathetic and parasympathetic divisions, which generally have opposing effects on the body's systems. The sympathetic nervous system prepares the body for fight or flight reactions, while the parasympathetic nervous system promotes rest and digest activities. The 2 main types of cells in the nervous system are neurons and neuroglia. Neurons transmit signals, and neuroglia provide support and protection. This image shows the structure of a neuron, including the cell body, dendrites that receive signals, and the axon that transmits them. Different types of neuroglia include astrocytes, oligodendrocytes, microglia, and ependymal cells, each performing specialized roles in the CNS. Myelin sheaths are lipid-rich layers that insulate axons, increasing the speed of nerve impulse conduction. Myelin is produced by Schwann cells in the PNS and oligodendrocytes in the CNS. The synapse is the junction between 2 neurons, or between a neuron and an effector cell, facilitating communication through chemical neurotransmitters. Neurotransmitters are chemical messengers that transmit signals across a synapse, such as acetylcholine, dopamine, norepinephrine, and serotonin. The cerebral hemispheres are the largest part of the brain and are responsible for higher-level functions, including thought, memory, and voluntary action. The brain is divided into 4 lobes: frontal, parietal, temporal, and occipital, each associated with specific functions. The frontal lobe is involved in personality, motor control, and reasoning. The parietal lobe processes sensory information like touch and temperature. The musculoskeletal system consists of the muscles, tendons, ligaments, bones, cartilage, joints, and bursae. Key functions of this system include locomotion, protection, blood production in the bone marrow, heat generation, maintenance of posture, and storage of minerals. There are 3 types of muscles in our body: voluntary skeletal muscle, involuntary cardiac muscle, and involuntary visceral smooth muscle. Muscles are composed of muscle fibers, which have numerous nuclei and striations, as illustrated here. In the intestine, smooth muscle forms 2 distinct layers that run along and around the organ. These layers create wave-like peristalsis, which propels the content. Skeletal muscle fibers are wrapped in connective tissue sheaths, including the endomysium, paramysium, and epimysium, which connect to the tendon and bone. This table compares skeletal, cardiac, and smooth muscle types, highlighting differences in location, appearance, cell shape, control, and function. Muscles have key properties: electrical excitability, contractility, extensibility, and elasticity, which allow them to respond to stimuli and change shape. In muscle physiology, muscle fibers are enclosed in sheaths. Each cell contains actin and myosin filaments arranged in a sarcomere, the muscle's basic structural unit. Muscle contraction occurs when actin and myosin slide past one another, shortening the sarcomeres. Calcium ions initiate this process, and ATP is essential for both contraction and relaxation. This diagram illustrates the structure of a muscle, showing muscle fibers, the motor neuron connection, and the arrangement of actin and myosin within a sarcomere. The structure of the sarcomere is defined by Z-lines and includes A bands, I bands, H zones, thick filaments, and thin filaments. Here is a detailed look at the neuromuscular junction, where a motor neuron connects with a muscle fiber to transmit signals. This illustration shows the process of signal transmission at the synapse, from the action potential reaching the terminal to the binding of acetylcholine and the initiation of a new action potential. Comparing relaxed and contracting muscle, the sliding filament theory shows that during contraction, actin filaments move inward, shortening the sarcomere and bringing Z discs closer together. As muscles contract, the H zone narrows or disappears, and the I band narrows, while the A band remains unchanged. Muscle contraction can be isometric, where the muscle length remains the same but tension increases, or isotonic, where tension is constant but muscle length varies. Muscle tone refers to the constant, low-level tension maintained by muscles. Muscles are also classified as fast twitch, which contract quickly and fatigue quickly, or slow twitch, which contracts slowly and resist fatigue. Smooth muscle is non-striated, contracts slowly, is autorhythmic, and is under involuntary control. Cardiac muscle is striated, autorhythmic, and also under involuntary control. Joint movements include flexion, which decreases the angle between two joints, and extension, which increases the angle. Abduction moves a limb away from the midline, while adduction moves it toward the midline. Additional movements include rotation, moving a bone around its own axis, and circumduction, creating a cone-shaped movement of a limb. Other movements are inversion, turning the sole of the foot inward, and eversion, turning the sole outward. Dorsiflexion points the foot up, and plantar flexion points it down. Supination rotates the forearm palm up, while pronation rotates it palm down. Protraction moves a part forward, and retraction moves it backward. Joints are classified by their structure and function. Fibrous joints are immovable, cartilaginous joints are slightly movable, and synovial joints are freely movable. Synovial joints are diverse, allowing a wide range of movements, and are characterized by a cavity filled with synovial fluid. Types of synovial joints include the plane joint, hinge joint, pivot joint, condyloid joint, saddle joint, and ball and socket joint. This diagram illustrates the structure of a synovial joint, including the joint capsule, articular cartilage, synovial membrane, and cavity. The nervous system is structurally divided into the central nervous system and the peripheral nervous system, and functionally into the somatic and autonomic systems. Neurons, the functional units of the nervous system, include cell bodies, dendrites, and axons, which are crucial for transmitting electrical and chemical signals. Neuroglia are supporting cells that provide structure, protection, and nutrients to neurons. Examples include astrocytes, oligodendrocytes, and Schwann cells. The nervous system generates and transmits action potentials through changes in ion permeability and membrane potential, following the all-or-none principle. A synapse is the junction between two neurons, or between a neuron and an effector cell, where communication occurs via chemical neurotransmitters. The central nervous system consists of the brain and spinal cord, which are protected by the meninges and cerebrospinal fluid. The cerebrum, the largest part of the brain, is divided into two hemispheres and controls higher functions like thinking, memory, and voluntary movement. The brain is divided into four lobes: frontal, parietal, temporal, and occipital, each responsible for different functions. The cerebellum, located beneath the cerebrum, coordinates voluntary movements and maintains balance and posture. The brainstem consists of the midbrain, pons, and medulla oblongata, regulating vital functions like breathing, heart rate, and sleep. This illustration details the major structures of the brain and their specific locations. The spinal cord transmits nerve impulses between the brain and the rest of the body. It consists of gray matter centrally located, surrounded by white matter. The spinal cord's gray matter contains neuronal cell bodies, while the white matter is composed of myelinated axons organized into ascending and descending tracts. The peripheral nervous system includes cranial nerves and spinal nerves, connecting the CNS to the limbs and organs. Spinal nerves are grouped into cervical, thoracic, lumbar, sacral, and coccygeal segments, each innervating specific body regions. The autonomic nervous system regulates involuntary functions through the sympathetic and parasympathetic divisions, controlling smooth muscles, glands, and the heart. The sympathetic division prepares the body for stress with a, quote, fight or flight response, while the parasympathetic division promotes rest and digest activities. This image highlights the anatomical differences and opposing functions of the sympathetic and parasympathetic nervous systems on various organs. This video provides a brief review of key anatomy and physiology topics for nursing. We will begin with a look at the paranasal sinuses, including the sphenoidal, ethmoidal, maxillary, and frontal sinuses. The vertebral column, or backbone, is composed of 32 to 33 individual bones. These include 7 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 3 to 4 coccygeal vertebrae. The vertebrae serve several vital functions. They support the weight of the head and trunk, protect the spinal cord, and allow spinal nerves to exit. Additionally, they provide sites for muscle attachment and permit the movement of the head and trunk. The cervical vertebrae are 7 in number. C1 is the atlas, and C2 is the axis. C7 is known as the cervical prominence. The atlas and occipital bone facilitate the yes motion, while the atlas and axis allow for the no motion. The thorax is made up of the sternum and ribs. The sternum has 3 parts: the manubrium, the body, and the xiphoid process. The sternal angle of Lewis, a slight elevation, identifies the location of the second rib. There are 12 pairs of ribs. True ribs are pairs 1 through 7, false ribs are pairs 8 through 10, and floating ribs are pairs 11 and 12. This image provides an anatomical view of the thorax, highlighting key structures such as the sternum, clavicle, ribs, and the angle of Lewis. The shoulder is constituted by the clavicle and scapulae. The clavicle articulates with the sternum and is the most commonly fractured bone. The scapula is attached to the ribs and vertebrae only by muscles and has an acromion process where the clavicle attaches. Here is an illustration of the bones of the shoulder, showing the clavicle, scapula, and humerus from both back and front views. The upper extremity is composed of the humerus, ulna, radius, carpals, metacarpals, and phalanges. These bones form the arm, forearm, wrist, and hand. This diagram shows the complete skeletal structure of the upper extremity, labeling the major bones from the shoulder to the fingertips. The pelvic girdle is composed of 3 fused bones: the pubis, ilium, and ischium. Together, these bones constitute the hip bone. There are distinct differences between the male and female pelvis. The female pelvis features a large, oval pelvic inlet, a shallow symphysis, and a broader sacrum. The male pelvis has a smaller, heart-shaped inlet and a deep symphysis. This slide visually compares the male and female pelvis, highlighting the structural differences, such as the pelvic inlet and the subpubic angle. This table summarizes the differences between the male and female pelvis across several areas, including the general structure, sacrum, pelvic inlet and outlet, and various bone features. The image displays the structure of the pelvic girdle, illustrating the connection between the hip bones and the vertebral column. The bones of the lower extremity include the femur, which is the thigh bone. The leg bones are the tibia and fibula. The ankle consists of tarsal bones, and the foot is made up of metatarsal bones. This diagram illustrates the anatomy of the knee joint, showing the femur, tibia, fibula, and key structures like the menisci and the anterior cruciate ligament. Cartilage is a dense connective tissue, consisting of fibers embedded in a strong, gel-like substance. It supports structures like the ear pinna and intervertebral discs. It also acts as a cushion and shock absorber. This image highlights the location of cartilage in an ankle joint, demonstrating its role as a smooth, cushioning surface between bones. This slide displays another view of the skeletal structure, likely focusing on specific anatomical features or joints. This detailed anatomical image likely focuses on a specific joint or area of the skeleton, possibly illustrating muscular or ligamentous attachments. The image provides an anatomical visualization, helping to reinforce the spatial relationships between bones and other structures. This visual aid further emphasizes the complexity and interconnectedness of the human skeletal system, particularly at joint sites. The image is another anatomical depiction, useful for reviewing the locations and forms of various bones and tissues. This slide presents another view of anatomical structures, likely continuing the review of bones and joints. This illustration offers a close-up or detailed view of a specific skeletal region for concentrated study. The visual content here serves to further solidify the understanding of anatomical relationships within the body. This slide provides yet another anatomical perspective, beneficial for thorough review in a nursing context. The final image in this sequence helps to conclude the visual portion of the skeletal anatomy review. The muscle system is divided into 3 types: skeletal, cardiac, and smooth muscle. Skeletal muscles are striated and voluntary. Cardiac muscle is striated and involuntary, and smooth muscle is non-striated and involuntary. Key characteristics of muscle tissue include excitability, contractility, and elasticity. Muscles are stimulated by nerves, can shorten and thicken to create movement, and can return to their original shape. Muscle movement occurs when a muscle contracts. The origin is the stationary or non-moving attachment point, while the insertion is the attachment point on the moving bone. There are different types of muscle contractions. Isometric contractions involve an increase in muscle tension without changing muscle length. Isotonic contractions involve a change in muscle length with constant tension. Fascia is a layer of connective tissue that surrounds the muscles. It can be superficial, deep, or visceral, providing support and protection to internal organs and structures. Tendons connect muscles to bones, and ligaments connect bones to other bones. Both are made of strong, fibrous connective tissue. This image illustrates the structure of the muscle, highlighting the muscle fibers, connective tissue, and the connection to the tendon. The body contains several types of joints, including ball and socket, hinge, pivot, saddle, gliding, and condyloid joints, each allowing for specific types of movement. Joint movement terms include flexion, extension, abduction, adduction, circumduction, and rotation. These terms describe the direction and type of motion at a joint. More specific movement terms include dorsiflexion and plantar flexion for the ankle, and supination and pronation for the forearm. This illustration provides a visual reference for the range of motion and different movements possible at various joints. The nervous system is responsible for controlling body functions and sensing the environment. It is divided into the central nervous system and the peripheral nervous system. The central nervous system includes the brain and spinal cord. The brain is the center for thought, memory, and voluntary action, while the spinal cord transmits signals. This diagram illustrates the structure of the spinal cord and the surrounding vertebrae, highlighting the central role of the cord in nerve transmission. The peripheral nervous system includes the nerves outside the brain and spinal cord, linking the central nervous system to the rest of the body. This video will provide a review of the anatomy and physiology of the nervous system. The central nervous system, or CNS, includes the brain and spinal cord. The peripheral nervous system, or PNS, includes nerves to the face, upper and lower limbs, as well as the ganglia. The peripheral nervous system is further classified into 3 functional divisions: the somatic nervous system, which controls skeletal muscles, the autonomic nervous system, which controls visceral organs, and the enteric nervous system, which controls GI tract functions. The central nervous system is composed of the brain, which consists of the cerebrum, cerebellum, brainstem, and diencephalon. The diencephalon includes the thalamus, hypothalamus, and pineal body. The brainstem is composed of the pons, medulla, and midbrain. This image highlights the major anatomical areas of the brain, including the frontal, parietal, occipital, and temporal lobes, as well as the corpus callosum, brainstem, and cerebellum. Here we see the diencephalon and brainstem, illustrating the thalamus, midbrain, pons, and medulla oblongata from anterior and posterolateral views. The cerebrum is the largest part of the brain, consisting of the right and left hemispheres connected by the corpus callosum. Each hemisphere has different lobes: frontal, temporal, parietal, and occipital. The basal ganglia are embedded within the cerebrum. This image shows the external surface of the brain, highlighting the major lobes: frontal, parietal, occipital, and temporal, as well as the cerebellum, central sulcus, and lateral fissure. The frontal lobe of the cerebrum influences personality, judgment, abstract reasoning, social behavior, language expression, and motor movement. The temporal lobe of the cerebrum controls hearing, language comprehension, and memory. The limbic system, located deep within this lobe, controls basic drives like hunger, emotion, and sexual drive. The parietal lobe is the principal center for the reception and interpretation of sensation. It interprets and integrates sensory inputs such as touch, temperature, and pain, as well as size, shape, distance, and texture. The occipital lobe of the cerebrum functions mainly to interpret visual stimuli. There are key speech areas in the cerebrum, Wernicke's area is responsible for the sensory reception of speech, and Broca's area is responsible for motor speech. This diagram illustrates the function of Broca's area for formulating speech response, the motor cortex for producing speech, and Wernicke's area for processing and comprehending incoming speech. This image provides a comprehensive view of the cortical functional areas, including the various motor, sensory, and association areas of the cerebrum. The cerebellum is the second largest brain region, also having two hemispheres. It functions to maintain muscle tone, coordinate muscle movement, posture, and control balance. Damage to the cerebellum results in decreased muscle tone and clumsy fine motor movements. This diagram illustrates the anatomy of the brainstem and cerebellum, labeling the lobules of the vermis and key brainstem structures like the midbrain, pons, and medulla. The brainstem lies inferior to the cerebrum and is continuous with the cerebrum and spinal cord. It is composed of the midbrain, the pons, and the medulla oblongata, and houses the center for respiration and the cardiovascular system. The midbrain connects with the cerebrum and contains numerous ascending and descending tracts and fibers. The pons connects the cerebellum with the cerebrum. It houses the respiratory and cardiovascular centers and serves as the exit point for cranial nerves 5, 6, and 7. The medulla oblongata is continuous with the spinal cord. It houses the cardiac, vasomotor, and respiratory centers and is the exit point for cranial nerves 9 through 12. The diencephalon is composed of the thalamus, hypothalamus, and pineal body. It is located superior to the brainstem and surrounded by the cerebral hemispheres. The thalamus is a relay station for sensory impulses to the cerebral cortex. It is involved in consciousness, alertness, and attention, and helps interpret pain, temperature, and pressure. The hypothalamus is the major regulator of homeostasis. Its functions include controlling the autonomic nervous system, regulating hormones, controlling body temperature, and regulating eating and drinking. The hypothalamus also regulates emotional and behavioral patterns, controls the circadian rhythm, and acts as a link between the nervous and endocrine systems via the pituitary gland. The pineal body or gland is part of the epithalamus and secretes the hormone melatonin. Melatonin contributes to setting the body's biological clock, influencing our sleep-wake cycles. The spinal cord is continuous with the medulla oblongata. It serves as a pathway for sensory and motor impulses and is the center for spinal reflexes. This diagram illustrates the internal structure of the spinal cord in a cross-section, showing the white matter and the H-shaped gray matter containing various horns. The gray matter of the spinal cord contains nerve cell bodies, neuroglia, unmyelinated axons, and dendrites, organized into posterior, anterior, and lateral horns. The white matter of the spinal cord consists of myelinated and unmyelinated axons. These are organized into ascending and descending tracts that carry nerve impulses to and from the brain. The nervous system is supported and protected by bone, meninges, and cerebrospinal fluid. The main structures are the skull, vertebrae, meninges, and ventricles. The meninges are three protective tissue layers that enclose the brain and spinal cord: the dura mater, the arachnoid mater, and the pia mater. The CSF is located between the arachnoid and pia mater. This image illustrates the location of the meninges, including the dura, arachnoid, and pia mater, and highlights the subarachnoid space where the CSF circulates. The four ventricles of the brain are interconnected cavities where cerebrospinal fluid, or CSF, is produced, circulated, and reabsorbed. They include the lateral ventricles, the third ventricle, and the fourth ventricle. This diagram shows the major ventricles, the two lateral ventricles, the third ventricle, and the fourth ventricle, along with the interventricular foramen and the cerebral aqueduct connecting them. Cerebrospinal fluid is a clear, colorless liquid that circulates through the subarachnoid space and the central canal of the spinal cord. It is formed by the choroid plexuses in the ventricles. CSF functions to cushion and protect the brain and spinal cord, circulate nutrients and chemical messengers, and remove waste products. It is constantly being produced and reabsorbed. This image illustrates the flow of CSF from the lateral ventricles through the third and fourth ventricles into the subarachnoid space and its eventual reabsorption into the blood. The blood-brain barrier, or BBB, is formed by endothelial cells of the capillaries and astrocytes. It is a protective mechanism that regulates the movement of substances between the blood and brain tissues. The BBB is selectively permeable, allowing oxygen, carbon dioxide, glucose, and water to pass, while blocking or slowing the passage of most other substances. Neurons are the functional unit of the nervous system, transmitting nerve impulses. They have three main parts: dendrites, the cell body, and the axon. Dendrites are short, highly branched fibers that receive input. The cell body contains the nucleus. The axon is a long, thin, cylindrical projection that transmits signals away from the cell body. This diagram illustrates the anatomy of a neuron, showing the dendrites receiving signals, the cell body, the nucleus, the axon transmitting the signal, and the axon terminals. Myelin sheaths, formed by Schwann cells in the PNS and oligodendrocytes in the CNS, cover some axons to insulate them and increase the speed of impulse conduction. The nodes of Ranvier are the gaps in the myelin sheath. Electrical impulses jump from node to node, which is known as saltatory conduction, increasing the speed of transmission. Let's review the parasympathetic responses. These include increased gastric and salivary secretions and peristalsis, and pupillary constriction. There is also decreased smooth muscle tone, causing sphincters to relax, and it's also responsible for erection. This table summarizes the autonomic nervous system. The sympathetic division, associated with the, quote, fight-or-flight response, has preganglionic cell bodies from T1L2. The parasympathetic division stimulates vegetative activities with preganglionic cell bodies in cranial and sacral nerves. In nerve physiology, nerve cells are excitable. A stimulus changes the membrane potential, generating an action potential for impulse transmission. The myelin sheath facilitates saltatory conduction, which increases nerve transmission speed. This illustration shows a diagrammatic view of neuronal tissues in the CNS, highlighting the relationship between neuroglia and neurons. Action potential conduction involves depolarization. The outside of the membrane becomes less positive, while the inside becomes more positive, moving the charge along the axon. Saltatory conduction is illustrated here, where the action potential jumps from one node of Ranvier to the next, increasing the speed of transmission along the myelinated axon. The synapse is the region of communication between two neurons, or a neuron and a target cell. A neurotransmitter is released from the nerve cell and binds to receptors on the target cell. This diagram details the synaptic transmission process. Neurotransmitters are released from the presynaptic terminal into the synaptic cleft and bind to receptors on the postsynaptic membrane, opening ion channels, comparing a normal reaction at the neuromuscular junction to the effect of a nerve agent. The nerve agent prevents the breakdown of acetylcholine, leading to sustained stimulation of the muscle. Next, we will discuss the special senses. Vision relies on stimulating photoreceptor cells, the rods and cones, in the retina. The eye has three layers: the fibrous layer, the UVEA, and the nervous coat, which is the retina. These diagrams illustrate the normal anatomy of the eye, showing key structures like the cornea, iris, lens, retina, optic nerve, and UVEA. Rods and cones are the receptor cells. Rods are crucial for vision in dim light, while cones are responsible for color vision. Both contain light-sensitive pigments, like rhodopsin. The process of phototransduction is a cyclical process. Light hits rhodopsin, causing a change that stimulates a cell change resulting in vision, and then the components are recycled. The optic nerve is a collection of fibers from retinal cells. It passes through the brainstem as the optic chiasm, and eventually reaches the occipital lobe for visual interpretation. This diagram illustrates the visual pathway from the eyes through the optic chiasm to the primary visual cortex in the brain. Light from the left visual field goes to the right side of the brain, and vice versa. Next, let's look at the vestibular apparatus, located in the inner ear. It is responsible for equilibrium. The saccule and utricle control linear motion, while the semicircular ducts control angular movement and acceleration. This illustration shows the components of the vestibular apparatus and its location relative to the cochlea in the ear. The hearing apparatus involves the cochlea and the auditory pathway. Sound waves are transmitted through the ossicles and converted into nerve impulses by the organ of Corti within the cochlear duct. The olfactory apparatus consists of the nose and the olfactory nerve. Stimulation of the olfactory nerves sends signals to the limbic system of the brain, influencing emotions and memory related to smell. This diagram shows the olfactory bulb and nerve pathways, illustrating how smell signals are transmitted to the brain. The gustatory apparatus, or taste, relies on chemoreceptors located on the taste buds, primarily on the tongue. There are 5 basic tastes: sweet, sour, salty, bitter, and umami. This illustration of the tongue shows the different types of papillae where taste buds are located. The taste buds, shown here, contain receptor cells that respond to chemical stimuli dissolved in saliva, generating nerve impulses for taste perception. The sense of touch, or somatosensation, involves mechanoreceptors, thermoreceptors, and nociceptors located throughout the skin and underlying tissues. This image highlights different types of sensory receptors in the skin, responsible for detecting touch, pressure, temperature, and pain. Next, let's review the main functions of the endocrine system. It consists of ductless glands that secrete hormones into the bloodstream to regulate various body functions. This diagram shows the locations of the major endocrine glands in the human body, including the pituitary, thyroid, adrenal, and pancreas. Hormones exert their effects by binding to specific receptors on or inside target cells, initiating a cascade of events that alter cell activity. Hormones can be classified as protein hormones, which bind to cell surface receptors, or steroid hormones, which usually pass through the cell membrane to bind to intracellular receptors. The hypothalamus plays a crucial role in controlling the endocrine system by regulating the pituitary gland. It secretes releasing and inhibiting hormones that affect anterior pituitary hormone release. The pituitary gland, often called the master gland, consists of two lobes. The anterior pituitary secretes hormones like GH, TSH, and ACTH, while the posterior pituitary releases ADH and oxytocin. This diagram illustrates the relationship between the hypothalamus and the pituitary gland. Hypothalamic hormones travel to the anterior pituitary and nerves project to the posterior pituitary. The thyroid gland produces thyroxine T4 and triiodothyronine T3, which regulate metabolism, growth, and development. Calcitonin is also produced here, regulating blood calcium levels. The parathyroid glands produce parathyroid hormone, which increases blood calcium levels, opposing the action of calcitonin. This mechanism is critical for calcium homeostasis. This illustration shows the location of the thyroid gland, situated in the neck, and the small parathyroid glands embedded in its posterior surface. The adrenal glands, located atop the kidneys, consist of the cortex and the medulla. The cortex secretes steroid hormones, while the medulla secretes epinephrine and norepinephrine. This diagram details the layers of the adrenal cortex and the hormones produced in each layer, such as mineralocorticoids, glucocorticoids, and sex hormones. The pancreas, a mixed gland, has both exocrine and endocrine functions. The islets of Langerhans secrete insulin and glucagon, vital for glucose regulation. Insulin promotes glucose uptake by cells, lowering blood sugar. Glucagon stimulates the liver to release glucose, raising blood sugar. This balance is crucial for energy supply. The pineal gland secretes melatonin, a hormone that regulates circadian rhythms, influencing sleep-wake cycles and seasonal functions. This illustration shows the key structures of the endocrine portion of the pancreas, focusing on the islets of Langerhans and their cells that produce insulin and glucagon. The endocrine function of the gonads involves producing sex hormones. The ovaries secrete estrogens and progesterone, and the testes produce androgens, like testosterone. The circulatory system is responsible for transporting oxygen, nutrients, hormones, and waste products throughout the body. It consists of the heart, blood vessels, and blood. Blood is composed of plasma and formed elements, red blood cells, white blood cells, and platelets. Plasma is primarily water, carrying proteins and other solutes. The parathyroid glands are important for calcium regulation. The hormone they produce increases bone breakdown by osteoclasts, increases vitamin D synthesis, increases calcium levels in the blood, and causes retention of calcium in the kidney. This diagram illustrates how parathyroid hormone, or PTH, works in calcium homeostasis. It promotes calcium release from the bone, reabsorption from the urine in the kidney, and promotes vitamin D formation to increase calcium absorption in the small intestine. The adrenal glands are a pair of glands resting on top of each kidney, with two layers, the adrenal cortex and the adrenal medulla. The cortex secretes mineralocorticoids, glucocorticoids, and androgens. The medulla secretes the catecholamines, epinephrine, and norepinephrine. This image shows a cross-section of the adrenal gland, highlighting its structure. The regulation of cortisol secretion is shown here. Cortisol from the adrenal cortex is involved in a negative feedback loop with the hypothalamus and pituitary gland. Cortisol's effects include increasing fat and protein breakdown, increasing blood glucose levels, and providing anti-inflammatory effects. Aldosterone, a mineralocorticoid from the adrenal cortex, is involved in the renin-angiotensin-aldosterone system, This system helps regulate blood pressure and electrolyte balance by affecting sodium and potassium levels. The adrenal cortex secretes mineralocorticoids like aldosterone. Aldosterone increases sodium and, secondarily, water retention and causes the excretion of potassium. Glucocorticoids, like cortisol, increase fat and protein breakdown and increase glucose synthesis. They also inhibit inflammation and the immune response. The adrenal cortex also produces adrenal androgens, including estrogens, androgens, and progestins. These are generally insignificant in males, but increase female sexual drives, pubic hair, and axillary hair growth. The endocrine portion of the pancreas is the islets of Langerhans. This islet is composed of 3 cell types: alpha, beta, and delta cells. This diagram illustrates the location of the pancreas and highlights the structure of the pancreatic islet, showing the alpha and beta cells. The alpha cells of the pancreas secrete glucagon, the beta cells secrete insulin, and the delta cells secrete somatostatin. This anatomical illustration provides a detailed view of the pancreas and surrounding structures, including various arteries and veins. Pancreatic insulin causes hypoglycemia through 2 main mechanisms: glucose breakdown via glycolysis and glycogen production via glycogenesis. Most body cells need insulin to allow glucose to enter the cell membrane. However, brain cells, intestinal cells, red blood cells, and islet cells do not require insulin for glucose entry. Pancreatic glucagon increases blood glucose levels by causing glycogen breakdown, known as glycogenolysis, and glucose production, known as gluconeogenesis. Focusing on the male gonads, the testes house the interstitial cells of Leydig, which secrete androgens, including testosterone, dihydrotestosterone, and androsterone. Androgens aid in spermatogenesis, maintain functional reproductive organs, and are responsible for male secondary sex characteristics and sexual drives. In the female gonads, the ovary, the follicular cells of the ovarian follicle secrete estrogen, and the corpus luteum secretes progesterone. Estrogen aids in uterine and mammary gland development, maintains the structure of the external genitalia, produces female secondary sex characteristics, and maintains a normal menstrual cycle. Progesterone prepares the uterus for implantation, sustains pregnancy, and aids in mammary gland development. The pineal gland, located in the epithalamus, produces melatonin, a hormone involved in regulating the sleep-wake cycle. The thymus gland, located behind the sternum, plays a vital role in immune function, where it helps T lymphocytes mature and differentiate. The thymus secretes thymosin, which promotes the growth and maturation of T lymphocytes, essential components of the immune system. The stomach secretes gastrin, which increases hydrochloric acid production and intestinal motility. The small intestine secretes secretin, which stimulates the secretion of water and bicarbonate from the pancreas. It also secretes cholecystokinin, which causes gallbladder contraction and bile release. The kidneys produce 2 hormones, erythropoietin, which stimulates red blood cell production, and renin, which is crucial for blood pressure regulation. The heart secretes atrial natriuretic hormone, or ANH, which opposes the effects of the renin-angiotensin-aldosterone system, leading to decreased blood volume and pressure. Prostaglandins are lipid-soluble hormones produced throughout the body that play roles in many functions, including pain, inflammation, and regulating blood vessel diameter. This video reviewed key concepts of the endocrine system, including the functions of various hormones and glands in maintaining homeostasis. Welcome to the next section of our video, a review of the musculoskeletal system, focusing on bone function and composition. The skeletal system provides support, protection, movement, mineral storage, blood cell formation, and energy storage. Bone tissue is composed of a cellular matrix. The matrix contains organic materials, primarily collagen and proteoglycans, and inorganic materials, primarily hydroxyapatite, which is calcium phosphate, and calcium carbonate. Bones contain 4 types of cells: osteoblasts, which build bone, osteocytes, which maintain the bone, osteoclasts, which break down bone, and osteoprogenitor cells, which are stem cells. Bone can be classified as either compact or spongy. Compact bone is dense and found on the exterior, while spongy bone is porous and found on the interior, containing red bone marrow. The basic structural unit of compact bone is the osteon, also called the Haversian system. Each osteon contains a central canal, lamellae, lacunae, osteocytes, and canaliculi. Spongy bone, or cancellous bone, is located on the interior of bones, has a latticework structure called trabeculae, and lacks osteons. It is where red bone marrow is located. This diagram provides a visual comparison of compact bone and spongy bone structures, highlighting the differences in organization. Long bones have several distinct parts, including the diaphysis, the shaft, the epiphysis, the ends, the metaphysis, the region where the diaphysis and epiphysis meet, and the articular cartilage covering the ends. The periosteum is the outer connective tissue covering of the bone. It contains nerve fibers, blood vessels, and osteogenic cells. The endosteum lines the inner medullary cavity. Bone development, or osteogenesis, begins in the embryo. There are two types: intramembranous ossification, which forms flat bones, and endochondral ossification, which forms long bones from cartilage models. The epiphyseal plate, or growth plate, is responsible for the increase in bone length during childhood and adolescence. When growth stops, it becomes the epiphyseal line. Bone remodeling involves bone resorption by osteoclasts and bone deposition by osteoblasts. This continuous process is essential for maintaining bone health and responding to stress. Hormones like parathyroid hormone, or PTH, and calcitonin regulate bone remodeling. PTH stimulates osteoclasts, while calcitonin inhibits them, thus regulating blood calcium levels. This concludes our review of the musculoskeletal system, focusing on bone structure, function, and development. This knowledge is fundamental for understanding physiological processes in nursing. This image displays a typical ECG waveform showing the P wave, QRS complex, and T wave, along with the PQ and QT intervals, which represent different phases of the cardiac cycle. Here we compare the action potentials of skeletal muscle and cardiac muscle. Note the distinct plateau phase in the cardiac muscle action potential, caused by the entry of calcium ions. The cardiac cycle consists of the contraction phase, called systole, and the relaxation phase, known as diastole, in each heartbeat. This illustration shows the heart's internal structure and valves during different phases. The image on the left shows the relaxed state, while the image on the right shows the contracted state. This diagram illustrates the full cardiac cycle, including atrial systole, ventricular systole, and diastole, highlighting the opening and closing of the AV and semilunar valves. Heart sounds, S1 through S4, can be auscultated. S1 is due to AV valve closure, S2 is due to semilunar valve closure, S3 is due to rapid blood rushing, and S4 is due to atrial contraction. Stroke volume is the blood pumped out per beat. Multiplying stroke volume by heart rate yields cardiac output. Cardiac output multiplied by total peripheral resistance gives blood pressure. Preload is the degree of stretching of the heart muscle when filled with blood. Afterload is the resistance the heart must overcome to eject blood. Starling's law of the heart states that contraction force is proportional to the stretching of cardiac muscle fibers. However, excessive stretching impairs contraction. This illustration details the cardioregulatory center in the brain and the role of sensory, parasympathetic, and sympathetic nerve fibers in regulating heart function and circulation. Blood vessels include arteries, veins, and capillaries, as well as lymphatic vessels. Arteries have thicker walls, are deeper, and carry oxygenated blood away from the heart. This diagram illustrates the structure of arteries, veins, arterioles, and capillaries, showing the flow of blood through the circulatory system and the different layers of the vessel walls. Veins are thin-walled, non-pulsating, and carry deoxygenated blood towards the heart. Arterioles and venules are small arteries and veins. Capillaries form a network connecting them. The diameter of arterioles largely determines peripheral resistance. Epinephrine, cold, and irritation cause vasoconstriction, increasing resistance. Histamine and warm temperatures cause vasodilation, decreasing resistance. Chronotropic effect changes the heart rate. Dromotropic effect changes the speed of conduction. Inotropic effect refers to changes in myocardial contractility. Myocardial cell types include myocardial cells, found in the myocardium, responsible for contraction and relaxation, and specialized cells of the electrical conduction system, which generate and conduct electrical impulses. Systemic circulation begins with the aorta, which leaves the left ventricle and branches. The vena cava returns blood from the whole body to the right atrium. This image provides a detailed visualization of the vascular system in the human body, illustrating the extensive network of arteries and veins. Blood pressure is the force exerted by blood against the vessel wall, measured by a sphygmomanometer. It's normally represented as systolic pressure and diastolic pressure. Pulse pressure is the difference between the two. Most exchange of gases and substances occurs through the capillary wall. This capillary exchange is primarily driven by filtration difference and diffusion. This image illustrates the structure and function of capillaries, highlighting their role in the exchange of materials between blood and tissues. This is an illustration showing the structure of an artery, including the tunica externa, media, and internal layers. This image details the structure of a vein, showing the layers of the wall and the presence of valves to ensure unidirectional blood flow back to the heart. Pulmonary circulation involves blood being pumped from the right ventricle to the lungs via the pulmonary artery, where gas exchange occurs, and then returning to the left atrium via the pulmonary vein. This visual representation shows the pulmonary circulation path, differentiating it from systemic circulation. Coronary circulation supplies blood to the heart muscle itself. The coronary arteries branch off the aorta and supply oxygenated blood, while coronary veins collect deoxygenated blood. This diagram provides an anterior view of the heart, highlighting the major coronary arteries and veins. The hepatic portal system transports nutrient-rich, oxygen-poor blood from the digestive organs to the liver before it returns to general circulation. This illustration details the path of the hepatic portal system, showing blood flow from the gastrointestinal tract and spleen to the liver. Fetal circulation is specialized to oxygenate blood via the placenta, bypassing the non-functional fetal lungs. Key structures include the umbilical arteries and vein, ductus venosus, foramen ovale, and ductus arteriosus. This diagram of fetal circulation shows the connection between the mother's and baby's blood supply via the placenta and the various shunts that bypass the fetal lungs and liver. Blood is composed of plasma-informed elements, red blood cells, white blood cells, and platelets. Red blood cells carry oxygen. White blood cells fight infection. Platelets are essential for clotting. Hematopoiesis is the process of blood cell formation, which occurs primarily in the red bone marrow, originating from hematopoietic stem cells. Erythropoiesis is the formation of red blood cells. It requires iron, vitamin B12, folic acid, and protein, and is regulated by the hormone erythropoietin. This image illustrates the process of hematopoiesis, showing the differentiation of hematopoietic stem cells into various types of mature blood cells. Anemia is a reduction in red blood cell or hemoglobin levels, decreasing the oxygen-carrying capacity of the blood. Leukopenia is a decrease in white blood cells. Thrombocytopenia is a reduced platelet count. Hemostasis is the process of stopping bleeding, involving vasoconstriction, platelet plug formation, and blood coagulation. Blood type is determined by the antigens on red blood cells. The ABO system classifies blood into types A, B, and O, based on the presence or absence of A and B antigens. The Rh factor is another critical blood group antigen. Individuals are Rh positive if they have the D antigen and Rh negative if they do not. This chart shows the ABO blood types, listing the antigens and antibodies present in each type, and which types can safely receive a blood transfusion. The lymphatic system drains interstitial fluid and returns it to the bloodstream, aids in immunity by producing lymphocytes, and absorbs and transports dietary fats. The lymphatic vessels are thin-walled, contain valves, and resemble veins. They converge to form two main ducts, the thoracic duct and the right lymphatic duct. The primary organs of the lymphatic system include the thymus, spleen, and tonsils. Lymph nodes filter lymph and house lymphocytes and macrophages. This diagram illustrates the human lymphatic system, showing the major vessels, ducts, and lymph nodes throughout the body. The respiratory system consists of the upper and lower respiratory tracts. The upper tract includes the nasal cavity, pharynx, and larynx, while the lower tract includes the trachea, bronchi, and lungs. Platelets, also called thrombocytes, are the smallest formed element in the blood with a lifespan of 8 to 10 days. They are involved in clot formation by forming a platelet plug in an injured vessel and releasing chemicals that activate the clotting mechanism.
Special thanks to London Heartbeat's Hom Academy UK for educational support, and request listeners to explore its 200 popular courses. Subscribe social media as Facebook, Instagram, YouTube for free regular updates, and not miss future amazing discussions. Subscribe to higher level of YouTube membership for early access to educational videos, and also even to have personal chats and guidance sessions.
This table summarizes the formed elements of the blood, including red blood cells, white blood cells, and platelets. It shows their illustrations, descriptions, and primary functions in the body. Blood types are grouped into A, B, and O, based on the presence of antigens on the surface of the red blood cells. Antigens A and B determine the specific blood type. If neither is present, the blood type is O. This table outlines the ABO blood groups, including the frequency in the U.S. population, the RBC antigens, corresponding plasma antibodies, and which blood types can be received. This diagram illustrates the relationship between antigens on the red blood cells and antibodies in the plasma for each of the 4 ABO blood types, A, B, AB, and O. Blood group A has antibody B. Blood group B has antibody A. Blood group AB has no antibody. And blood group O has no antigen but contains both antibody A and B. Blood typing involves testing blood samples with anti-A and anti-B sera. Agglutination or clumping with a specific serum indicates the presence of the corresponding antigen, allowing determination of the ABO blood type. The Rh system exists alongside the ABO group. The D antigen is the most prevalent Rh antigen. A person with the D antigen is Rh positive, and one without it is Rh negative. This image shows a blood test card used for typing blood, including tests for anti-A, anti-B, and anti-D serums to determine the full ABO and Rh group. Rh incompatibility can be a problem if an Rh negative mother carries an Rh positive baby. Exposure to Rh antigens during the first birth causes the mother to make anti-Rh antibodies, which can endanger a subsequent Rh positive fetus. If a mother is Rh negative and the fetus is Rh positive, fetal red blood cells can enter the maternal circulation. The mother's body produces anti-Rh antibodies, which can cross the placenta and cause the agglutination of fetal red blood cells, leading to hemolytic disease of the newborn. Thank you so much for watching this video.